Oh no, the bacteria are surrounding us. Olivia, what do we do?
Don't worry, I just found a cheat code that should help us out.
Look, they're getting stronger. I think they're adapting.
Quick, let's change our environment and get out of here.
Well, as our game has just reminded us, while the discovery and development of antibiotics has revolutionized healthcare, we often find that when humans mess with nature, the results don't always turn out as we had planned. The environments that bacteria live in can be harsh, and in order for bacteria species to survive in these conditions, they have to adapt.
Many of these adaptations come about through mutations, while others can arise when bacteria exchanges DNA with another bacteria. This is where AMR, or antimicrobial resistance, comes into play. Antimicrobial resistance refers to the variety of adaptation mechanisms that are used by bacteria to survive an encounter with antibiotics or other toxic substances. So for the most part, our cheat codes will not work.
AMR occurs naturally and can take place no matter where a bacterium is located. It starts in the environment, where natural compounds called antibiotics are made by bacteria and other organisms like fungi. These naturally occurring antibiotics allow organisms to colonize or establish dominance in different habitats, especially in places where nutrients are hard to come by. As the environment transforms over time, bacteria are able to quickly evolve alongside changing conditions. Resistance to antibiotics can be expressed either intrinsically, meaning it occurs independently from previous antibiotic exposure, or by selective pressure, which occurs because bacteria are exposed to an antibiotic and most adapt to survive.
Bacteria not only have natural resistance, but are also able to gain resistance by sharing DNA with each other. We call this process horizontal gene transfer. This means that bacteria not only inherit DNA from their mother cells, but they also can share DNA from nearby bacteria, or even pick up pieces of DNA directly from the environment. So what this means is that bacteria are able to share antibiotic resistance genes, or ARGs. The genes can be found in many places, like on bacterial genomes, on circular strands of DNA called plasmids, or as free-floating genetic material. The specific ways of how bacteria interact, resist, and survive in the presence of antibiotics are complex. For example, there are many different mechanisms for bacterial resistance, including a strong outer membrane layer that provides a shield to certain types of molecules, the modification and changing of the actual antibiotic when it enters the cell, and efflux pumps that actively remove toxic molecules, including antibiotics, from the cell.
While naturally resistant bacteria are present everywhere, this source of resistance is not supposed to be a global health problem. In fact, resistant microorganisms and animals have existed together for thousands of years. However, humans need antibiotics for a number of reasons. We use antibiotics to fight diseases, such as veterinary pathogens, to improve livestock production, and to even protect crops from bacterial plagues. For these reasons, many decades ago, humans began to produce and use a massive amount of antibiotics. Although the antibiotics we make might be similar to the ones produced by bacteria, they can also be new chemical compounds that are not found in the environment. This antibiotic era caused a dramatic increase in selective pressure on environmental bacteria, causing them to rapidly develop and acquire resistance to the antibiotics they are directly exposed to, driving the spread of AMR in clinical and veterinary settings, as well as in the environment.